Welcome everybody to SynBioBeta Live. My name is John Cumbers and I'm the founder and the CEO of SynBioBeta. And we're here inside the virtual world of Nanome having a pretty awesome COVID-19 party today. There's not much to party about the virus, but today we're going to go deep inside the heart of the virus, looking at the protein coding shell, looking at the RNA inside of it, and particularly looking at the spike protein, which is the part that interfaces with the receptor on the human cell surface. And we're going to be looking at some of the antibodies that bind there and what people are doing with the antibodies. If you're just joining us, then welcome. This is SynBioBeta Live, and my name is John Cumbers. I'm the founder and the CEO of SynBioBeta. We'd love to know who is on the call today. Normally we use Zoom for doing these town halls, but today we're actually live on YouTube. So instead of typing in your questions and comments into the Zoom chat, we want you to switch over to YouTube and type in your comments and questions into the YouTube chat. And Kevin, our producer, is online, and he's going to be reading out your smart and sophisticated questions and answers in the comment box. So please tell us who you are, where you're dialing in from today, and we'll do a shout out to you shortly. I'm joined today by four amazing guests who I'm going to high five each as I introduce them. Robert Scoble is the Scobleizer. He's the VR guru, and he's been understanding virtual worlds for many years and the impact that VR is going to have on our lives. And so very exciting to have him join us today. And after we delve into the science, I'm going to have a fireside chat with Scoble and with our next guest, Philip Rosedale. Philip Rosedale is the founder of the VR virtual world Second Life. Phil, thanks for joining us.
Hey, good to see you.
And so Phil is here today really to help us understand the intersection of virtual worlds and biology and what we can see happening in the future in terms of everybody working from home right now, what the next few months might look like in terms of virtual reality and virtual worlds. So Philip, thank you for joining us as well.
Thanks, good to be here.
Next to my left here in the beautiful green Captain Planet glasses, we see Steve McCloskey, and Steve is the founder of Nanome, and that is this virtual world that we're in. And Steve, if you could just give everybody just a couple of sentences on your academic background and your credentials to be a molecular engineer, that would be great.
Yeah, definitely. So I'm Steve McCloskey, and thanks, Sean, for having me here. So I'm a nano engineer, and as a nano engineer, here at the nano scale with all sorts of chemicals, like this is my home. So really, everywhere between a single chemical and a full virus is the nanoscale. So like 1 nanometer to 100 nanometers. And
awesome.
We're the first nano engineering department in the world at UC San Diego. So still a lot more tech to come.
Fantastic. So before I introduce our fourth guest, we're going to play a little quiz for you folks at home. I just want to grab each one of these molecules here and show everybody what they are, but I'm not going to tell you what they are. What I'd like for you to do is to be able to tell me what they are. So I'm going to hold up this first one here. Hopefully you can see that. And for those of you who haven't tried this tool, I'm using the Oculus Quest headset, which is a wireless headset. Everything is contained within the Oculus. My colleagues here on the left and right of me all have desktop versions, so much higher powered graphic machines, which is what we'd recommend if you're going to get deep into this. But for me, the amateur structural biologist, this is a pretty amazing experience. And I tell you, once you put this on, the world of biology and the world of biochemistry and the world of chemistry completely changes. So I do encourage you to check this out. We're going to be doing a lot more things with VR over the coming months. So this is the first test.
John,
yes.
Could you tell the audience what the blue and the gray and the green are? Maybe that gives them a little hint as to what this molecule is.
I could help out. So the grays are all carbons, the blues are nitrogens, the whites are hydrogens, and this green right here is chlorine.
Ooh,
oh, that's a big clue as well to what this molecule is. So here's molecule number one. Type it into the YouTube box if you know what this molecule is. And I'm just going to pass this. If you could just hold that for me a second. Thank you. The next molecule is going to be this one. And I should probably put them both together so that you can, because that's kind of part of the magic here. So we got these. This was 1 on the bottom. This is 2 on the top. So if you can type them into the comment box if you know what these two molecules are. And then something smaller, oops, something smaller here.
Grab the other two. I have a few things hidden behind this that we're going to go over next. So yeah, just grab these down and place them next to this guy.
Got it.
There you
okay.
Go. Yeah.
There we go. So then we have a third molecule here at the bottom. I'm just gonna, yeah. So.
You could grab the whole thing up by grabbing the bottom
great.
one. Or yeah.
Got
perfect.
it. Okay.
There you go, John. You're a pro.
Thank you. So here's this one. And my favorite party trick, I'm going to grab this one. So again, type it into the YouTube Messenger if you know what it is. I'm going to grab this one. Can I separate it and just grab that bottom one or not?
Yeah, sure. Here, I'll just unlock it real quick.
I just want to show my favorite party trick, and I've done this with my 8-year-old daughter and my 6-year-old son, which is to grab this carbon ring here and check it out.
Oh, yeah. When you two-hand, it does everything because it scales everything relative. But if you do one hand, it'll be independent.
Got it. Okay. But if I scale, it'll do everything?
Yeah, because everything scales together. Because you don't want like one big chemical and one small chemical. Everything is relative.
Well, my main party trick is doing this, like a giant ring, and I hope you can see that. Our cameraman today is Keita. Thank you very much, Keita. So I am now dancing in this giant carbon ring. Scientifically not that important. Coolness factor, 10 out of 10, Steve. Absolutely. Party on, Captain Planet. Okay, so I'm going to now introduce our fourth guest, who is Rob Rhinehart. And Rob is with the coronavirus project, and he's also with Mars Bio, which is a venture fund in LA. Rob, if you could just introduce yourselves and tell us a little bit about the coronavirus project.
Sure. So at the Corona Initiative, we're contributing to research and development efforts around the virus. So we support basic science such as structural biology, crystallization of proteins whose structures are missing, as well as research into different vaccines and treatments, and development efforts as well around new diagnostics, new research assays, and new delivery methods and peptides and proteins for therapeutic routes. And we'll be talking about a few of those today. So thanks for having me.
Awesome. Thanks for coming.
And John,
yeah.
We should have talked about my intro before, but that's okay. I just finished a book called The Infinite Retina, which is on spatial computing. And I wrote that with Rena Cronin, who's my partner at Infinite Retina Consultancy in the space.
Awesome. Thank you, Scoble. I appreciate that. And what we also now want to see if Kevin, if you can tell us if anybody won the competition in terms of these structures.
Yeah, we had a couple of guesses. A few folks guessed hydroxychloroquine or chloroquine, hydroxychloroquine. A few people got that. And there was a guess from Jim, thymine on one of the molecules, and Joy said that the third molecule is adenine. How close were folks?
Well, I'm going to pass these back over to Steve, and Steve, over to you, and you can see
cool.
Tell people how they did.
Yeah, thanks.
Also, a guess for hula hoop carbon ring.
Cool. Well, congrats to the people that guessed this one as chloroquine, and this one as the hydroxychloroquine. And you can notice like they're pretty much the exact same, except for this oxygen here. So if we wanted to, make one of these into a, hydroxychloroquine, we're really just adding a little oxygen right here.
No way.
Yep. And then now they're the same molecule. And, we could also add the hydrogen, or that could just be autofilled. But yeah, we, full periodic table, we could, just kind of like build with anything that we want, and just add that little hydrogen there. Yeah. Now they're exactly the same. So, yeah, so 2 hydroxychloroquines, and this one was a cytosine.
So hold on 1 second, Steve, before you. And I'm hearing a bit of background. I'm not sure who that is, but Steve, if you could just explain the significance of chloroquine right now.
Yeah. So I would love to actually show you guys how this works, but the scientific community, there's no consensus on how this is supposed to work at the nano scale. The only evidence that we have that it actually treats COVID-19 is from doctors prescribing it and treating patients. So it's really hard to say when we don't have like particular molecular targets and understand the exact mechanism of action, if this is effective or not. But according to doctors, some doctors around the world, it may be an effective treatment. And we know that it's effective against things like malaria already.
Now, there is a theory to the nanoscale mechanism, which is that the pH is affected, which in turn affects glycosylation of the ACE2 receptor, which is the viral entry mechanism, which we're going to look at in a second. And so there's limited evidence, but there is a theory as to the mechanism involving glycosylation of that receptor, which in turn affects the binding affinity of the receptor binding domain of the spike protein. So that would be a good experiment to run in vitro.
Cool. Yeah, and we could show you what those two pieces are, but you basically have like two proteins that combine, and if this caused one of those proteins to get extra chemicals on it, then it might hinder the binding of that.
Great, excellent. Okay, so if you want to, we'll take these two away and focus on the other one. What was excellent? Yeah, do you want to explain about?
Yeah, and so let's go ahead and just show a DNA backbone here. So what we could do is, so typically when people look at DNA, people talk about sequencing all these COVID-19 samples around the world and like how they're changing based upon who gave it to who. You kind of get different like substrains of it going on. Well, that's all encoded by RNA or DNA. So right here, we have the typical like double helix that we would all see, in front of us. But what's actually going on is we have molecules, real chemicals going on. So we could look at that here, and we could see that this is just, one of the chemical groups that's placed right there. So if we were to just, hide that guy real quick, you can see that this is just one of the base pairs. And what's really cool about looking at these base pairs is that they all interact exactly with each other. So you have this one accepts another hydrogen, this one donates its hydrogen. This one accepts the hydrogen, this one donates it, and then same with over here. So you have that like complementary action, and all of that is what causes these two halves to find each other. So yeah, just a quick little explainer before we go into a lot of the details. Yeah.
And we don't normally we do a poll when we do the Zoom, and we ask how many people are beginners, intermediate, and advanced. In this case, we don't have that functionality in YouTube, so I'm not sure. But I just want to keep everybody up to speed. If you could just bring up the periodic table again.
Yeah, definitely.
So just a brief history of time. Big Bang, contraction, expansion created all of these different elements on the periodic table. I can't remember how many there are in total. How many are there, Steve?
118 plus or minus.
118.
I mean, yeah, people kind of, the last few are kind of strange because like they're highly unstable. Like we don't even put them on because you're not going to build a chemical with them ever.
Got it. So this is the palette, and biology really only uses is it 6 of them, schnapps?
Yeah, carbon, nitrogen, oxygen, hydrogen, some phosphorus in there, sulfur in there. Yeah, so kind of like this whole right side.
And so if what we normally do when we look at DNA, if you could take off the detail structure, what we normally see is a shape like this, where you just got an abstract representation of the double helix. But what Steve was just going into then is if you turn on the molecule view again, each one of these balls now represents an element on the periodic table, and you can match the colors. So there's oxygen in red, there's oxygen in red here, there's nitrogen in blue, there's nitrogen in blue here. And so now if we go back to then what you were saying about that structure that you pulled up and we asked everybody what it was, if you could just repeat that one more time.
Oh yeah, the cytosine?
Yeah. So there we go.
Yeah.
So you can match that to the periodic table. You can see exactly which elements are on here. And now if you can put it back into the structured DNA, you can see how our world was constructed from all of these different 118 elements. All right. Excellent. So that is one base pair. So and you said it was cytosine, is that right?
Yeah, cytosine.
Which is the letter C. So the opposite letter over here would be the letter G. And that's it, folks. At the nanomolecular level, we can see exactly what's going on. and abstraction is some key important element of being able to bioengineer as well. Great, Steve. So I'm going to pass the rest over to you, and we're going to take a deep dive through what Nanome can do, what this virtual environment can do. People can go to Nanome and download this environment themselves and explore. So we're going to see what it can do, and then we're going to bring up the coronavirus and see how the structure of that looks like.
Okay, perfect. And yeah, just wanted to mention to our viewers at home, like you might see like this is two-dimensional representations. And what we do in Nanome is we actually run a energy minimization on these in order to get the three-dimensional structure. So, this might be how you draw it, but in reality, everything is three-dimensional. And so you see it pop out there. It's using just intermolecular force fields that go between all the atoms. And that's what causes it to take up different angles where you have, this popping out, that's pretty planar. And of course, with Nanome, you could also make your own chemicals. So we'll get to a little bit of this later, but just to show, like earlier we were drawing hydroxychloroquine. Let's just say we wanted to keep modifying things. You could really modify things however you want. And if you actually make a mistake, like right now, this nitrogen already has a double bond and a single bond. If we had a fourth, or too many, really, it just is going to turn red like that. And so that's how we know that we don't want that, and we could just undo all those actions. So, yeah.
So who's here thinking, who in the room here is thinking they wish they had this in chemistry class? Right? I mean, this is just phenomenal.
I might not have gotten into journalism if I had this in chemistry class back 35 years ago.
I mean, this is seriously amazing what you've done here and what you're enabling, Steve. So kudos to you. This is just such an amazing tool.
Cool, thanks, Sean. And yeah, so we're going to be diving into some protein structures next. And I just wanted to say that when you look at the DNA here, this DNA is going to be encoding information, and so every 3 base pairs is going to be 1 amino acid, where 1 amino acid is essentially 1 part of this protein structure here. And we currently have it in a wire view, so it might be a bit hard to see on the stream. But what I wanted to show was just these blobs. So the mechanism is that it goes from the DNA encodes proteins, and then we actually need to solve the proteins. And the way you do that is by looking at blobs. And so if you see all these blob shapes, they have very particular things that they're encapsulating. And what scientists do is they actually fill in all the coordinate pieces, the balls and sticks, in order to build up the protein structure. And you'll see that over here, it fits pretty much exactly perfect. Yeah, so we get the blobs through X-ray diffraction. Oh, it's John on the Wi-Fi. Okay, I think he'll be coming back soon. Yeah, so you get the blobs by expressing your protein in a solution or in a species like yeast or E. coli or cell cultures, and then you purify it and you crystallize it, and then you bombard it with X-rays and diffract them. And then you're able to see this 3-dimensional blobs. This is really experimental data you're getting.
So cryo-electron microscopy is another method for larger proteins and complexes.
Yeah, and if you have a really large protein, then you can't crystallize it. Then what you do is you just shoot electrons through it, and then actually capture images that way. And then you could reconstruct those images into a 3-dimensional map as well.
And I just want to make sure that we haven't lost people along the way. We're not going to go in and explain the central dogma in terms of how DNA gets converted into proteins, but central dogma is something that you should search on YouTube and watch some videos about. But generally, DNA is the 2-dimensional long string of A's, C's, T's, and G's, and it gets converted in a 20-dimensional alphabet of amino acids, which creates structures, which are the things that are the shape, the physical representation of biology, enzymes that can make and break other chemical bonds. And that's now what we've moved from DNA, talking about DNA and A's, C's, T's, and G's, to now talking about these beautiful protein structures made of the 20 different amino acids. Steve, I actually got a little bit lost between this blue structure and then the ball and stick structure behind. Are we looking at exactly the same thing, or you're just moving this box over the stick structure and seeing the representation in the
yeah, well, so this cube, this box, this could go on forever, pretty much. Like yeah, the way that we're looking at this is like a refinement of a certain type of space, and so this is all going to be like symmetrically repeated because it's a big crystal. And so yeah, so in theory, like this should be another protein that looks just like this guy. And then when we go over, we see that this part has been filled in.
Got it. Okay, great.
Yeah.
Thank you. Please continue.
Yeah, and there's also some like, we could vary the sigma values. Like this is the probability density of anything being there. So we could say that, this has a, there are more refined atoms here because it's, the atoms are very likely to be in these spots. But if we go like closer to 0, it just becomes noise.
So this is traditionally done in a program called Coot. Is that right?
Mm-hmm. Yeah, Coot's pretty popular. Paul Amesley is the developer there. He's a really great guy. Pretty much all the industry uses his free software. So yeah, all these proteins pretty much come from Coot.
And CCP and CCPI2 is commonly
Yeah.
used in determining these structures from X-ray data.
Cool. Yeah, so we could take a little pause here in case there were any questions, or we could move on to some of the SARS.
I hope people watching at home on YouTube understand how incredible this environment is. That when you're in VR, the acuity of these molecules and atoms and chemical structures is just incredible, and you can walk around them, and then if you give me control, I can grab them and zoom them in and really feel like I'm right on top of the molecule. It's amazing.
Go really for it. Yeah, you got control. Just to actually, that box is kind of independent from the rest, by the way.
Oh, that's okay. Sorry.
Scoble, what does acuity mean?
It just the way it feels to you, the visual, which is presence and immersion, but the visual sharpness of the molecules is much higher than what you're able to see on YouTube. It feels like you're actually touching a real molecule or a real atom.
It also makes the structural biologist's job much easier. So with that map that Steve was just showing, a lot of those molecules have to be filled in by hand. Some of them are done by software, but some of these massive proteins do have to be filled in and built up manually. And a structural biologist has to know which atom and which bond is going where, and being able to interact with it in this way makes that job much easier and much more intuitive.
So this has been a lot of fun doing this, and we'd like to bring in more scientists to experience Nanome and to experience this virtual world. So we had George Church on our town hall yesterday. We're going to get George into this virtual world soon. Jake Glanville at Distributed Bio is working on an antibody therapeutic, and so he wants to come in and show us how his antibody docks with the coronavirus. If you have other scientists that you think we should be inviting who we want to experience this, then please type them into the YouTube chat box, and we'll probably build up a whole series of these that we can do in VR. Kevin, any questions or comments from the audience that you want to give us? Just feel free to shout them out as we go.
Yeah, well, let me just jump in there. A few comments. Kambiz Hamadani just kind of remarks that the ability to synchronously display and manipulate molecules in 3D in different labs across the globe will be transformational for drug discovery efforts. And a lot of people are just saying that's really incredible to be able to explore proteins like this. There are a couple of general... Yeah, sorry?
I was just saying, thanks, Kambiz.
Oh, yeah. And
shout out.
There are a couple of general questions, like John Westra asks, how long will it be before we go from VR, AR, nanoscale molecular design to application simulation and ultimately automated 3D assembly of nanoscale objects? What role will AI have?
Okay.
And,
I think those are great questions that could segue us into looking at the protease structure, actually, of the SARS-CoV-2.
Yeah.
Yes.
Okay, I'll load that up.
And it's also thinking about the design build test cycle here and how these tools are going to come into place. How quickly after we look at a drug, an antibody that docks to the virus, how quickly can we actually then synthesize the DNA and get that antibody expressed and see whether it does exactly what we are predicting it would do.
Cool. So over here, so this protein is actually the protease, the main protease that is a protein that can like break apart and cleave other proteins. And the way that it does that is actually through this binding pocket here. And there's been a lot of efforts from both traditional medicinal chemistry to design a better molecule, as well as artificial intelligence. Yeah, sorry, Philip, I kind of had you covered there.
I was in it.
you have some COVID on your shirt.
Yeah. That's wonderful. Just enjoying it. Yeah.
What does it smell like, Philip?
Yeah, nothing I can tell you.
It's
the one thing we can't do in here.
Well, one of my friends had it already, and one of the first symptoms is you lose your taste and smell. So you might not be able to smell it if you had it on your shirt.
Right.
Interesting.
Oh, yeah. So we could go through some of the features of this structure. So this is the original 2003 outbreak version of SARS. And there's a few areas highlighted in this orange. And so all those areas highlighted in orange are actually the differences between in the beige, we have the original 2003 SARS, and then in the blue, we have the new SARS-CoV-2, which causes the COVID-19 disease.
And this is the spike protein that we're looking at, is that right?
Actually, we could do the spike protein next. This one is actually the protease first. Just kind of starting small, and then the spike is really big, so we'll save the best for last.
For kids at home who don't know what protease is, can you define that for us?
Yeah, so a protease is a type of enzyme. And so what this does is kind of like a factory inside of the virus that's chopping things up and getting things ready, helping the virus function how it needs to function. And Rob, I know that you've been working on some science stuff. Anything you wanted to add on a explainer basis for the audience?
Is that remdesivir in that pocket right there?
I don't believe this is remdesivir in the pocket. But yeah, people talk about remdesivir being a potential treatment, repurposed drug. And yeah, this would theoretically be the target for remdesivir.
Mm-hmm.
And remdesivir is a small molecule drug. I believe it's Gilead's drug. Is that right?
Yeah. So remdesivir is a small molecule drug. We could actually pull it up if we want. We could just type in remdesivir. Oh, looks like I already had it typed in. Yeah, so this is remdesivir in the two-dimensional mode. For the most part in the databases, they're stored like this, so we need to run that minimization. There we go.
And again, for those people who are new to drug discovery, the term small molecule sounds like just some random descriptor. It's actually pretty specific. It means it's a class of drug. That means it's a chemical like this, which is defined as a small molecule, as opposed to something like an antibody. Many drugs are now antibodies, which are much larger molecules with more diversity. Well,
there
I guess similar kinds of.
There are also peptides that are somewhere in between.
Yeah, peptides, small protein chains.
But this is definitely a small molecule.
So a question for me, some people are on the internet are saying this is a human designed virus. The scientists say it's not. How would you tell the difference between a human designed virus and one designed by nature?
So some things would be like immediate giveaways. So right now all these orange parts are the differences. And it looks like these could be naturally occurring differences. That is, there's a bunch of mutations in the amino acid sequence, and the way that you get those is from mutations in the DNA or the RNA. Mm-hmm. And so if there's not too many, it seems likely that it could just have evolved. But a dead giveaway, which scientists have not seen, and that's why the scientists are saying it probably wasn't engineered, would be like Frankensteining, parts together and like adding some, viral vector, some viral RNA over to this one, and then like combining parts from other viruses. And if that happened, it'd be very obvious that it was engineered. What is more subtle here is that there aren't really, really obvious modifications like that. It's kind of just mutations here and there. And what's interesting, what's interesting here is that none of the mutations were actually in the binding pocket. All of them were just sort of peripheral on the side. So it's like a highly conserved, active center is what it's called.
It would seem the probability through random trial of basically finding this number of small mutations would be infinitesimal, right? I mean, so.
Well, I mean,
but nobody could make it.
The mutations, do seem kind of random.
And I was listening.
However, it makes for such a more infectious virus. Some of the important mutations we'll look at in a second on the receptor binding domain, and you'll see even though the mutations seem that it seems like there's just a few of them, and they seem to be at random, and yet it leads to a binding affinity of 10 to 20 times higher. Which allows it to get into cells that much stronger and ends up being a far more infectious virus than SARS 2003.
Yesterday, I was listening to the Stanford AI COVID presentations. They had a conference yesterday, and one of the presenters said that there's about 8 different strains already of this virus, and that it's mutating every 2 weeks. Does that match to what you're seeing? And where did you get this version of it? And can you bring in new ones as scientists discover new mutations as well?
So this version actually came from the RCSB Protein Data Bank, which is like a big online repository where scientists deposit their structures. So both of them actually came from there. And what happens is RNA viruses mutate pretty rapidly. So there could be like minorly different strains happening all the time. Like that does make sense. And if we don't have a full crystallized protein structure for all of those, what we can do is homology modeling. And so if we look at this sequence menu over here, this sequence menu shows you the sequence of amino acids. And we could see that there's differences like that S and that A, and then everything. Oh, yeah, sorry, here, let's, how about we make it, okay. Maybe there for all of us?
Yeah.
Okay, cool. All right, and so hopefully that's better for everyone. And yeah, we can see N and S. And whenever I click on these, it's being highlighted in the structure as well. And so you can see that this one is a little bit obscured behind the blue there. Others are maybe a bit more prominent. V, K, L. Those might be on the backside. Yeah, those are on the backside over here. And so this just maps out the differences. And if we had a new strain, like let's just say there was a third column going on here, and this sequence is nowhere in any way similar. But we can map a third sequence and also then see what's going on with the new strain compared to the original SARS over on top, and then the new COVID-19 version on the second line.
Got it.
Steve, I just want to make sure that we haven't lost anybody along the way. So now what we're seeing on this menu up here, instead of A's, C's, T's, and G's, this is now an amino acid alphabet of 20 different amino acids. And I just want to highlight, looking at this image, if you could just toggle off just show us the original SARS virus. So this is SARS-CoV-1, which was the original SARS virus, and this is what it looks like. If you could just make it a little bit bigger.
The protease.
I'm sorry. Yeah, this is so we'll come back to the protease in a second. So this is the protease from the original SARS-1, and now if you can, and why are we seeing ribbons and ball and stick? Oh, the ball and if you could just help me understand that.
So the ribbon gives us a good idea of like the total structure, and then the ball and stick is really just around this active center, the binding pocket that we're interested in, and it's all the amino acid residues that are close to that. And we could also see the surface view as well. And with the surface view,
so you can toggle on and off different parts of the giant model to show different kind of representations, different levels of abstraction, depending on what you as a scientist are trying to do. In this case, you're interested in this part here, so you're able to change the structure so it doesn't look like this ribbon structure. Instead, it's like this stick structure, which will help you.
The surface. You could turn off the ribbons and just have a bunch of sticks or balls and sticks, or we could do.
Wow. Okay, go back a second. Let's just one. Okay.
That's pretty cool.
I mean, this is pretty amazing. Just that you're just swiping through all these different models like it's child's play here. But I mean, this is just pretty fantastic. All these different representations of the elements in the periodic table.
Steve, can you go back to surface view?
Yeah.
So I just wanted to point out, if if you were doing something like drug discovery, you would note that this binding pocket here makes sense to target. And so, and this is conveniently where remdesivir fits in pretty well. So, if you're developing a drug and you're going after the protease, you can look at the entire surface and think about what might make sense for a small molecule to target. A small molecule would fit very well potentially into this pocket, or you could design a peptide or an antibody to try to go after epitope on the surface or a larger area. But because this is happening inside the host cell, having a small molecule fit into this binding pocket, thereby disrupting the action of the protease, would be a very attractive target for a drug. And we can see that remdesivir actually does fit very nicely into this pocket, thus disrupting the function of the protease, thus potentially acting as an effective therapeutic.
And Rob, just by look, sorry. Quick clarify.
I was just going to ask, when we see pictures of the virus in media, it tends to be a round thing with a lot of red spiky
right.
things.
So this is not the entire virus. So the virus and what you're looking at would be the capsid.
So the virus would be like
on envelope
all around it.
It is roughly spherical, and then there are 40 to 50 of these spike proteins sticking out. And we'll look at the spike protein in a minute. But the spike protein is important for cell entry and host fusion. So what happens is the end of the spike protein, this area called the receptor binding domain, which can be either closed or open. It will open and then fit into the ACE2 receptor, which kind of holds it in place. And then there's an area on the spike called the fusion domain, which ends up fusing with the host cell, and its RNA enters into the host. From there, it starts to transcribe its called an ORF, an open reading frame. And so the RNA encodes several viral proteins, which it will use to copy itself. And the protease is a really important one because this reading frame actually generates a very, very kind of large globular protein, which is kind of multiple proteins linked together. And then this protease, an ase means that it's cutting something. So this protease will actually cut the those linkers of the giant protein apart, leading to individual proteins which have individual functions, such as creating new virus particles. And so if you were to disrupt this protease, it would not be able to do that job. It would not be able to segment out the individual virus proteins, which means that you would just have this non-functional large virus-related protein, and it would not be able to function. It would not be able to create new virus particles with that. And the idea is eventually the host cell would break that down, present it on the surface, signal the immune system, and hopefully generate a better response against the virus.
Wow.
Well, yeah, it's a perfect.
It's amazing that this. Why does nature design a virus? How did this happen in nature?
So
what's the goal of the virus? What is its goal? It's to propagate, right?
To survive and replicate, and all organisms do that.
Interesting.
Yeah. So, when it binds, to a human cell, if that RNA could get inside the cell, then the cell starts producing the right proteins, and then, all those proteins self-assemble into a new virus. And then that virus goes on to live its life. So it's just a continual cycle of life.
Yeah, and this is not what the virus looks like in your bloodstream or saliva.
Right.
This is only active after the virus enters the cell. So really, it'd be better to catch it before it gets into the cell. So if you're designing a drug, you might think, okay, this is good. If you're already infected, potentially this is going to treat you. But the virus is already in the host cell. So it would be better. And what if you don't get all of the proteases? Another protease might continue to do its job even though you've blocked this one. And so these are all considerations that would go into designing a therapeutic.
So I just want to make sure everybody caught up with your detailed description, Rob, which I really appreciate. What we're saying here is that this is a protease. A protease breaks up other proteins. This is part of the virus payload. Once it gets into a human cell, it releases its RNA. This protease is made, and this then goes and wreaks havoc on your human cells. Is that right?
Essentially, yes. This is a very important part of the functioning of the virus, of its replication, of its activity inside the host cell.
Great. And Steve, if you could just now go back to the two different views where you were showing the original SARS virus, and then if you could show we're showing the original SARS virus in one color, And then if you could show the new SARS virus in another color. And what you're seeing when you're seeing these ribbons now, so if you could just talk me through that. Yeah, so turn on one of them, which is the original SARS virus, which we'll see in one color.
Yeah. So in this, like the ribbon view with the beige with the original SARS and then the blue with the new CoV-2.
Perfect. So I just wanted to point out to folks that that's what we're seeing here. The reason these are multicolored, like blue and beige, is because that's where we're overlapping two amino acid sequences, and we can see that they match. And as Steve pointed out earlier, then the big orange parts are the highlights. That's where it's different, and that's how structural virologists can look at this and say, aha, this is different, and that helps us understand why our immune system can't recognize it, because there are different parts, and also how we might then target antibodies and vaccines and therapeutics against it because of the different parts.
Got it.
And if you could just then, I had another. Oh, Rob, you said about looking at a protease and that binding pocket. Can I just control it for a second?
Yeah, sure.
If you look at that binding pocket, that was here. Is that right? This is the binding pocket.
Look in there. Yeah. So if you're looking at it, and you're designing a drug against it, of the whole surface, that is probably going to jump to the top as what you would want to target as a binding pocket.
And by binding pocket, we're talking about a peptide, another protein going in here and the protease cleaving it. Is that right?
Now, because this is so small, it's going to be difficult. You could, in theory, but it's going to be difficult to design a peptide or an antibody to go after that. And now keep in mind, antibodies are often active in the bloodstream, and this is inside the host cell. And so in many ways, it's kind of late to get an antibody against it. Perhaps you could do a peptide, but remember, then you need to get that peptide inside the cell. And so.
But what I'm curious about is, what does this naturally bind? Is it a protease binds a peptide and chops it in two? Is this the place where a peptide would go?
Virus, I'm not 100% sure.
Yeah, I believe this is the active center of the protease.
So any enzyme is going to have an active region. I think Steve's saying this is the active region on the protease. And
got it.
So
and that's why we now
blocking that. So if you put a molecule in there instead, it's not going to be able to do its job.
And this is how drugs work. This is, you find a protein that's doing something, it's causing havoc, chopping up your proteins inside your body. Stick a drug in here, it can no longer function.
That is one approach. I mean, really, anything you can do to disrupt the functioning, you could cut this in half. You could cover the entire thing. You could denature it. You could change the pH. There's many approaches, but a pretty simple and elegant one would be to have something that would not be toxic to your cell, would not be toxic to your body, and it would not have off-target effects, but it would fit into that binding pocket, and which is also the active region of this enzyme, and prevent it from doing its job.
Topologically,
it's a question from the audience. Frank Diane asks, is the binding pocket, is that where the catalytic, the question is the binding pocket is where the catalytic triad? It's kind of an incomplete question. Does that make sense?
I want a catalytic triad.
Steve, do you know what he's asking?
I'm not exactly sure. So I'm not completely an expert on drug design. I work with a lot of pharmaceutical and biotech companies that are working on drug design. So active triad.
I mean that is a catalytic action, though.
Yeah.
I mean it's splitting of the larger virus protein. So
I believe this is the active center. Yeah, this is the active site of the protein.
So we're not sure, but if anybody else knows, type it into the chat box. Any other questions, Kevin?
Now keep in mind this is not steady too. Like the whole thing is kind of vibrating. Everything is moving. Maybe we can show a simulation later, but this is not static. It is not exactly like this in shape all the time. And so one thing you need to consider is how long is your molecule going to stay there? Is it going to stay there forever, or is it going to, more common, you see things bind for a set period of time? And there's equipment where you can kind of measure all this in vitro or kind of in the lab.
Awesome.
Around binding affinity and kinetics, and there are different tools to measure proteins binding to proteins and molecules binding to proteins. And so another thing you would look at is how stable is this interaction? How long is this molecule going to stay in that pocket?
Fantastic. So I want to do a time check. It's 8:45. I know we promised everybody the spike protein. Steve, anything else we want to discuss here, or can we move on to the spike
Philip,
protein?
if you had any comments, and then we could move on to the spike.
I was just going to say this is such a great example of how VR, and again, as we mentioned, you really can't see this on Twitch. My background's physics, and so the only thing that really stands out to me is the concavity of this region, right? Which I'm sure is an example of a physical intuition. I'm not a biologist. That's easy to gain from being in VR. You can see that the situation here is that a lot's going to get stuck in here, which is what you guys are saying, if I understand correctly. And it's fun how you look at that.
Yeah, this is remdesivir. This is the Gilead drug here.
This is actually not. Sorry,
I'm sorry.
This one's just one that I made last week and submitted to a crowdsourced chemical design competition being run by UK Diamond Light Source and John Codera's lab in New York. So they're actually synthesizing the molecules that were submitted last week. They're making them this week.
Oh my goodness,
exciting.
That's fantastic. We're definitely seeing a lot of excitement, a lot of news. Just something in the New York Times I saw yesterday, which talks about how scientists are collaborating at light speed, all on a common thing. I mean, I've just learned in the last 3 weeks so much about virology and immunology and now structural biology. So that's really
amazing. You made a whole new drug with a very strong, it's very possible. It's a really strong mechanism of action. This drug was designed in a week and made in the next week. That pace of discovery and development is unprecedented.
If we had a live studio audience, I could say, you get a drug, you get a drug. I guess we do have a live audience, so you all get drugs at home for you YouTube followers.
You can make your own drug at home.
Cool.
Steve,
so I'll load up the spike next then.
Show us the virus.
Yeah.
Why can't the audience hear you, Philip? Do you know? Oh, okay. Okay, sorry.
Yeah. So there's a few things going on here, and I'll just
Yeah. So this is a bigger protein. It's going to take a minute.
So as a reminder, this is live, and if you've got Q&A for us, you can type it into the YouTube comments box, and we'd love to take any questions or comments. And if you want to experience this for yourself, you can go to Nanome. Is it nanome.com, Steve? Is that your website?
Not nanome.ai.
Nanome.ai, and you can download this. And I'm on a Oculus Quest headset. I'm running a little bit slower than everybody else in the room. That's just the way my life is. But I will be. But if you want to get into this deep, then you want a PC with a cable to the headset so that you can run as fast as these guys.
If you have an Oculus Quest, you can get a link cable to an NVIDIA or an AMD graphics card that's in a.
That's what I use.
PC.
Yeah, if you have a good kind of desktop graphics processor using the link cable, the Quest is very good. You can see very large proteins. It works really well even without a Rift.
Yeah.
Awesome. And so what Steve is doing
we haven't talked about VR overall. There's environments like this for car design, architecture. There's a whole bunch of things that are happening in VR, and we're going to talk about that shortly.
Cool.
That's awesome.
Yeah. So I just pulled up an image. I believe this image is of the coronavirus, and we could see on the edges here we have all these spikes coming out.
Extreme close up.
And this one's actually an electron microscope image, and so this is like real experimental data captured on a microscope versus over on the right that's a huge just like computational model.
Wow, was this made by Rami?
I'm not sure. I saw this actually posted on social media last night and was like, oh wow, like that is a great explainer for how the spikes come into play in relation to the entire virus.
And then inside you can see the RNA, and is that the nucleocapsid?
Yeah, exactly.
So the deformation of its structure is the interaction with the environment and with the other.
Well, what they did here is they actually just took the viruses and put them on a TEM grid, transmission electron microscopy, and then froze them using cryogenic freezing. And so it's called cryo-EM, and then they, shoot the electrons at them and then collect them. And yeah, there's all sorts of things that could happen with the viruses getting deformed just during processing and sample prep and naturally occurring things as well.
And can somebody help me with the scale here compared to a pinhead? How many of these viruses could we fit on a pinhead? Does anybody know?
So these are about 100 nanometers in diameter. It varies. I think it's maybe between 100 and 120. Maybe some are smaller, like 80 nanometer diameter. And also to give you a sense of the RNA. So coronavirus is about 30 kilobases, which is
30,000 base pairs.
Yeah, which is actually a little large. HIV is around 10,000.
Got it.
I think
so that would be about
BSV is around 10,000.
I can't do the math on this. Is that about how many on a pinhead would that be if it's
I would estimate about 1 million on a pinhead.
Wow.
And it could be more because these viruses could all it's not just like flat all on a surface of a pinhead. Like they could stack on top of each other. So
yeah, they can be very dense. I mean, I've seen measurements of concentration in saliva between 1 and 100 million per milliliter.
Yep.
Amazing. Great. So now what we're going to do is zoom in on one of these spike proteins, these red and brown ones. So again, we're going to zoom right into one, and then you're going to see at these different scales, again, going back to the periodic table, then how many individual elements are involved in one of these. It's quite remarkable how, particularly in VR, we can see the scale and zoom in and zoom out like this.
Yeah, real quick, I just wanted to make a note. I brought in another protein unrelated to SARS or CoV-2 or anything, but this is just a molecular dynamics simulation being played back. And I just wanted to emphasize that, everything is moving, all these proteins over here, like they're all moving. This isn't a static object that's completely, fixed. All these little spikes are wiggling, all the inner parts are wiggling, all the surface parts are wiggling. Some parts wiggle more, some parts wiggle less. And
this is just Brownian motion that we're looking at. This is just.
Yeah, we're looking at so Brownian motion, I believe, is for larger particles, and this is just molecular dynamics. So this is treating every sphere as if it is just like a rigid ball that has a connection between every other sphere based around different interatomic potentials.
Awesome, amazing.
So yeah, we could go ahead and start interacting with some of the spike protein structures. I'll just get rid of that. All right. So yeah, this might be a little bit large for you, John, but I think everybody else should be able to see the spike. Yeah. So one of these spikes, I'm just gonna go ahead and put it on the image there. One of these spikes is one of those spikes. So like, you literally think about like all these things hanging off the surface. That's a spike, spike, spike. There's like going to be
maybe 50.
Yeah, dozens of these, maybe 50. And that's also over here as well. Like, we see that all these little like blobs, hanging off the end. Those are the spike proteins. And the reason it's called the coronavirus is because it looks kind of like a crown. You have all these spikes hanging off the edges. So they said, oh, it's like a crown. Let's call it the coronavirus. When they first discovered these things. And so now, we're just going to interact with our spike protein structure here. And there's actually a really important part of the spike protein. This is what Rob was talking about, the receptor binding domain, or the RBD. And so we could just look at, just this guy just to clean everything up. And we'll pull up the human ACE2. Actually, we could do a comparison as well between the original SARS, which is in blue, and the new SARS-CoV-2 in the green.
And so this is important because this is a big reason why this is so much more infectious.
Exactly. Yeah. All the mutations that occur, and we could also pull up the sequence menu again. All the mutations that occur between the original SARS RBD and the COVID RBD. We align those together, and we could see that there's going to be several gaps, and then also some mutations. And these might actually just be both from COVID, actually. But we could also import other ones if you guys wanted to do a comparison. The main things I wanted to show was actually how it binds with the human protein, which is ACE2. And I've cleaned it up.
This ACE2 is on the surface of a host cell.
Mm-hmm.
and it's usually
So that is,
not used for this, right? The virus is hijacking this receptor as an entry vector, and it doesn't go all the way through this receptor. It binds to it because the spike protein is very large compared to the extracellular domain of ACE2, and so that's how it attaches to the cell. And then there's another part of the spike which is called the fusion domain, and that then kind of falls down onto the membrane of the host cell and then merges with it and then inserts its RNA.
Awesome. So Rob, Steve, I want to be respectful of everybody's time. I know that Philip has a meeting to get to in 5 minutes in the real world. I know that all of us can stay on a little bit longer to chat more about this spike protein, which I'd like to do. But before we do that, as we're about to lose Philip, Philip is the founder of Second Life. Maybe you can describe the enormity of Second Life and the impact that it had on virtual worlds. And then maybe you and Scoble, having been living this world for 10 or more years, can help us understand the impact that you think virtual reality is going to have both on the biological sciences like this, and then maybe other expert domains. And then what are your predictions over the next 3, 6, 12 months about how life's going to change because of the whole world in lockdown?
Well, that's a whole lot of stuff to answer, Jim. But
in 5 minutes,
4 minutes.
4 minutes.
Yeah, exactly. Maybe, hold on, hang on. I mean, a few thoughts. Second Life, as you said, was a virtual world before there were any of these headsets. So since I was a kid, I'd wanted to build a big virtual world. And so I got a chance to do that starting in 1999, actually, when I started the company of Second Life.
Holy cow, 21 years ago.
Yeah, and it's still going strong. Second Life is still a big world with a lot of people in it doing a lot of different things. But it existed before there was this ability to put a headset on and see things in 3D like we're doing it here. So I think there's a moment of transition here where we're starting to get this ability to actually get these fantastic intuitions that we can stand around a molecule and look at it and get a sense of something like this binding to the ACE region and understand what's really going on. And that really is the promise of full immersion in these virtual worlds. I think the other point to be made here is we're also seeing the front line of where VR headsets, such as they are, can really help us right now. This is a scientific meeting, and we're kind of making the very best use of this new technology. With luck, VR headsets and VR technology can actually help humanity with something like this. And I'm really excited to see that transition happen. I think that social use of these headsets is still a bit on because they're not comfortable enough to wear all day, and you can't send email while you're doing this, to some benefit perhaps, but there's still a challenge in getting to everybody wearing these things every day. But I think we're sort of in a time machine right now where because of the urgency of the moment and because of the specific way we're using this together really gives us an advantage. So I'm just blown away being here and getting these intuitions about this structure. As I said, my background is in physics. It's not anything as complex as this, and I'm able to ride along with you guys and understand, and that's pretty inspiring.
Yeah.
Thanks for having me here. Yeah.
The science behind VR is real interesting. Jeremy Balanson at Stanford gave me the same demo he gave to Mark Zuckerberg of walking across planks on floors and talking about how he's able to really change the brains of people, for instance, who have had a stroke or other mental illness. And there's real exciting science on that front. Irina and I have been writing a book about 7 industries that are going to be radically changed over the next decade because of this kind of technology, from manufacturing, where like Volkswagen has virtualized its entire factory floor. So instead of walking around a molecule, we could be walking around a Volkswagen factory right now and talking about that. And so, we're seeing Scoble, many changes coming. Right.
If you can just pause 1 second, we got to say thank you, Philip. We'd love to talk more and involve you in future events. Thank you for
absolutely
for
really great to see you guys. Thank you
Thanks,
very much. Yep.
Thank
see
you
you soon. All right, guys.
Thanks. Scoble, if you could just tell us what
it's funny that took me back. Philip and I were walking around with Mark Zuckerberg about a decade ago talking about sharding databases and at Davos and hanging out for a couple hours. So it's really great to see Philip again.
It's awesome, and it was kind of fun. We did a demo of this yesterday to prep for this, and this was the first time that I've really had a virtual meeting. And I talked about it yesterday on a town hall with George Church. I mean, how phenomenal this is. There's a bond, there's friendships forming, there's professional relationships. Steve, you and I have never met. Scoble, we've just met a couple of times. Philip, I just met once. Rob and I are pretty close friends already, but there's a bond, right? I mean, this is way different than a Zoom.
Oh yeah, with everyone working from home, I've really been encouraging people to just do their meetings, just do their conferences.
And I think Phil, to be honest, after 21 years of this, I think Phil is a little bit jaded. I'm looking at my 6-year-old son making friends in Rec Room, and
cool.
Which is an online environment for gaming. Oh my goodness!
My son loves that.
Yeah, we should have our kids.
The real problem is very few people have headsets, and Oculus Quest has sold around 700,000 units so far, which sounds fairly impressive. Wow. But when you compare it to a mobile phone, where 2 billion people are on mobile phones, right? It's a very small number of people.
Well, at some point, only 1 million people had mobile phones.
It's true.
They also haven't. Yeah, they haven't seen the YouTube stream of this yet.
No, I've been telling people to get into VR for years, and most people ignore me. But now that we're in this new world of staying at home and working, people are starting to understand the power of virtual reality. Philip has been beaten down by the markets a bit, and it's funny. I talked to Doug Engelbart, who's the guy who invented the mouse and all sorts of things, right? He was in the room when the internet was first turned on, the first nodes between, I think it was UCLA and SRI where he worked. And he was angry at the end of his life, sort of like Philip gives us a little like, I'd love to do this every day, but it's just not here yet. And it takes time for these things to happen, and it takes technology to keep shrinking. We're not there yet. These headsets are heavy. They make you sweat. They cost money, right? My system costs around $2,000 that I'm playing with you here. An Oculus Quest is $400, which is really much more affordable. But in the next few years, you're going to see more new technology shipped from the technology industry than humans have ever shipped in our history. So think about that, what's coming in this next wave, which actually the virus, I think this thing has sped up. I was expecting this process to take a decade. I think the next process that we're going to go through as human beings is going to happen much faster because of the virus.
Awesome. So I want to come back to the virus, but just before we do, Scoble, in your new book, what are the 7 industries that are going to be disrupted?
If I can remember them all. FinTech, retail is radically changing. If you go to an Amazon Go store, you can go and pick up a product and leave without talking to a cashier, and that's using spatial computing technologies. Transportation, in fact, Irene and I are probably going to be interviewing Sebastian Thrun for our book launch in May. He's the guy who ran the Google self-driving car team and now is working at Stanford and is running Coursera and other things, and is doing flying vehicles, all with this new technology. Manufacturing is radically changing. Like I said, Volkswagen has its entire factory floor virtualized, so we can train workers faster using these technologies. Healthcare, obviously, this has deep impacts on healthcare. The ability to come up with drugs and new treatments for PTSD and Alzheimer's and dementia. We talked to Skip Rizzo, I think, also at UCLA, and he's doing a bunch of research about that. What else did I miss? There's a few others. Education.
Well, I think
this has deep impacts on education, right? Could you imagine every school kid in the world having access to this kind of visualization and this kind of teaching from experts, right? With their local teacher sitting next to them. It would be amazing, right? We're years away from that because of the cost of these things, but you can see that it's coming, right?
Yeah. Well, I think if you're publishing it in May, there's still time to add synthetic biology as the 8th industry, and you should interview Steve and Rob for the book.
No, we included that in healthcare because I think this is an important part of healthcare: the science, the ability to come up with new things and understand how the chemical and biological world works, and inspire new people to join this world and work on problems. I mean, didn't you say you just put a drug into open source? Right?
Yeah, we made a drug, put it out online to the public, didn't patent it, just gave it out there.
So that's real exciting, right? Because this is a new kind of machine that you're working on and you're trying to hack it. And so you're going to have virus hackathons probably in an hour, right?
I mean, you still need to bridge that with the real world. You still need to make what you designed, and
absolutely
for a molecule, that's going to be a different story than from a peptide, which is going to be a totally different story than from an antibody. And even in here, designing a whole antibody, which is possible. Actually, Steve and I have been working on designing whole new proteins in here. But you still need to connect that with the real world, and any structural biologist will even tell you that you never really know until you run the real world assay. You still need to get the actual materials. You still need to run the chemical experiments, and so this is a very useful tool. But we're not leaving the real world behind anytime soon.
No. In the mechanical world, you have a lot of 3D printers and things that you would design something in this space and then print it out on a 3D printer. How would you print out that drug? Tell us a little bit about the physical process that you would take this molecule you designed and then try seeing if it works in the real world.
Well, yeah. I mean, again, depends what you're making, but for a molecule, you're synthesizing it.
So for a molecule, you might start with a known chemical and then start adding on different pieces. And so imagine we started here, and then we wanted to add a amide. Every time you're adding something to it, you're going to be building this chemical even larger. And so you could just keep making it with like more and more like chemical building pieces. And then depending on like what order you do it, or sometimes you could even do just one beaker type of like all-in-one type of solution. So it really depends on what chemist is involved, or how you plan on synthesizing it. And there's also programs out there that'll use like machine learning and other tools in data science, where once you design the three-dimensional blueprint in VR in a 2D screen, the program will figure out many different paths on how to synthesize it, and then give you suggestions and say, oh, if you only have these reagents, here's how you would do it. But if you have these other reagents, then you could do it this way.
And one way to synthesize it, even something like this, and John will love this, would be synthetic biology. This is what a lot of people in synthetic biology do. You could have a chemical that's very expensive to synthesize with traditional methods, or some of the reagents are very hard to come by. But life and cells are synthesizing chemicals all the time. These are beakers. These are reactors. These do amazing things with very basic components. So you could make this with a cell. You could make this with different enzymes. And if you're making it an antibody or a protein, I think this is just the extracellular domain of ACE2, and this is just the receptor binding domain of spike. You could make these in cells. You could make the whole ACE2 receptor, and people are doing this. You could make the whole spike protein. You could make it in E. coli. You could make it in insect cells. You could make it in hamster cells. You could make it in human cells. All
so that's a really good
possible.
That's a really good
synthetic biology, and in fact, a lot of people are doing this, and this is a crucial part of researching this virus, is that people are quickly spinning up these cell lines and making some of these proteins that people are studying in new hosts.
That's a good point.
This is really interesting. If you were, I'm sure there's some high school kids who are going to watch this. What do they need to study for the next 4 years, going to a university, to be able to put themselves in play, to really have an understanding of how all this works, and really be able to add, a solution to the next pandemic, because there'll be another one someday. And there's a second war that we're fighting at the same time. We're fighting a war against this thing. We're fighting a war against climate crisis, climate change.
Yep.
And so, thinking about how do we take carbon out of the air, or how do we de-acidify oceans, or how do we fix some of the environmental problems we're causing.
I would think
those are very joined.
I would say get your hands busy now. I mean, I think running actual experiments is the best way to learn. And this is a wonderful tool right here. I've learned so much myself using this tool, and I think
but there's some basics here that you have to understand of how
the
chemical world works, how the physical world works, and how the biology works that we're seeing here. I don't have the skills to go and
studying studying biochemistry, biology, immunology, cell biology, and I'm a big fan of Coursera personally. There's a lot of free courses online I've gotten a ton of value out of, but again, I'm the biggest fan of actually just doing experiments. A lot of these materials are completely safe to work with. There's a website I really like called the Odin. You can buy everything you need to do some cell biology experiments for sometimes under $100 or a couple $100, and with a sink and a refrigerator, you can practice on a lot of these techniques, and it's perfectly safe. I think running those actual experiments and doing you can make yeast glow, and he even has a mammalian cell engineering kit right now. It's definitely within reach of a home lab or someone just eager to learn. So I really encourage people to do some experiments.
Yeah, Scoble, if I could chime in here, I actually turned my mic on for the audience so that they could hear me. So hi everyone, I'm the camera. So how's it going?
Hey kiddo.
You're running for people who
yes, I'm running the camera, and I'm using a keyboard and a mouse. But I really wanted to chime in here because I have a little bit of a media background, and the way I taught myself like basics of digital media was by going through tutorials of Photoshop and Final Cut, and Premiere and all those types of things. So personally, my hope with Nanome as a co-founder of Nanome is that middle schoolers and high schoolers can teach themselves the basics of organic chemistry, and material science and all those things through the software, so that that and have the software be that easy to use. So that's kind of one of my passions as well. And I think on the climate change side, Steve, I would love to hear you talk about kind of some of the initiatives. Everything's are made of atoms, including the inorganic side, right? So some battery research is actually happening using Nanome as well. And Steve's background is actually in material science, so that's something that I wanted to quickly chime in about.
Thank you, Kate. I appreciate that. So, Steve, I'll pass back to you, and just doing a time check. Maybe we do another 5 minutes after I pass back to you, and then we can wrap up.
By the way, we didn't cover for kids who are watching. How much does this environment cost? And can they try it for free? Can they start doing everything we're doing here? But tell us how they get started.
So they could do pretty much all this for free. All these are public structures. They could download public structures for free. There's a paid version that's more for like the academic researchers and people like that, where you could import custom structures, like ones that you design in other applications. You could just import them in. But yeah, if you wanted to get in here and start, playing with proteins, peptides, small molecules, all that's included in the free version. And then if you join a free public room, there's actually like a round robin mode that we have that switches presenter between different people to help make you share. So there's just a few things like that, but we'll give you trials for the paid version if you'd like to experience the full feature set. We know everything going on with coronavirus, people are stuck at home. We want them learning about proteins and peptides, and we want them to be able to self-educate either through our YouTube videos or our tutorial series, or just some of the content that we're seeing being published by the community, because we have a lot of academics that are doing research, that are also teaching, that are creating their own customized content as well.
Great.
Very cool.
And I want to do a plug for the tutorial. When you load Nanome, there's a 20-step or so tutorial that you can go through, which is also fantastic. Also, if biotech companies want to license this and use it, what's the best way for them to get in contact with you, Steve?
They could just email hello@nanome.ai. We'll be pretty quick to respond, get them set up. Biotech, pharmaceutical companies, typically they're more protective of their data. not really just sharing it whole world like us right here. And so there's a little bit more involvement with getting them set up for that.
But you can do that, and you have biotech companies
we could do that. Yeah, so they could just send us an email, and then we'll get that sorted out. But they could try out the free version with just public structures already if they wanted to as well.
Great. So just one thing before I pass back to you, Steve. Scoble, you asked about how do you go about making these things. Well, if there's an antibody, for example, a much larger molecule that you've decided fits beautifully in this structure, and you want to design it and you want to make it, there are a whole bunch of synthetic biology companies out there that will synthesize the DNA for you. It's very cheap now, just a few cents per base pair per A, C, T, and G. And a lot of the companies that sponsor SynBioBeta are the DNA synthesis companies. So there's companies out there like Twist Bioscience or Synthetic Genomics DNA or GenScript, many other companies that synthesize DNA. And you could send them a structure, and they could send you actually back the DNA. Depending on the size of it, it could be overnight if it's just a small oligo, or it could be within a week or so if it's a larger gene. And there are other companies out there, contract research organizations, that will then make that antibody for you, or you could express it yourself if you have the equipment. So it's pretty easy now to close that design build test cycle, and then now bringing in this whole VR element to it where you can actually visualize what's going on. It's a brave new world and a very exciting new world.
Yeah, and Scoble, I've taken actually a portion from our ACE2. So if you notice, these are both like the same exact thing. So what I did is I just selected it and then duplicated it, took that out. And so this is a human protein in the yellow. But what we could do is we could just snap off a portion of that human protein, and now we just have a peptide chain. And what a group at MIT is doing is they're actually using this as a blocker to say that if we just make this peptide, we might be able to actually bind to the spike protein over here, because it has all the same components that would bind, and then that would prevent the ACE2 protein from getting in there. And we could actually just hold that back a few frames. Yeah. So like if you have something already blocking it, then that guy is not able to come in there.
Decoy human.
Yeah.
Wow.
And in theory being, because the RBD is so strong at binding this ACE2 receptor, one of the best things to block it would be that binding region itself.
Yeah. Yeah. And in terms of, then synthesizing it, so the way that we would express this is we have 27 amino acids here. I think in the paper for MIT, they had like 30. But yeah, we start with serine number 1 down at the bottom. Then we go all the way to leucine number 27. And so that means we're going to have 3 times as many base pairs, because every 3 base pairs accounts for just one of these.
Yes.
And and then you just take that sequence of, my math is 81 base pairs. And then you express that in your yeast or E. coli or cell line, whatever is going to be producing it. And then that'll cause it to produce a bunch of these.
That's amazing. Wow.
Yeah, or actually what the group in MIT is doing is they actually have more of a synthesis pipeline where they could mix amino acids together in like a chemical fashion and assemble peptides that way. So peptides are kind of the hybrid. Like proteins over there, like if it's really big, you definitely want to express it in a biological entity like cell line, yeast, E. coli. But then peptides, you could make kind of like a chemical, because it is really just like a very large chemical here.
Yeah.
We could turn off the parts that make it look more like a protein and turn on the parts that make it look like a large chemical. So yeah, here it is. It's just a large chemical.
And again, those colors are representing the elements of the.
This would have made chemistry class so much more fun.
Steve, do we have
back
time?
to all the pain I had of trying to learn, how chemistry worked back in college, and it was one of the hardest classes I ever took, and many kids failed, and this would bring a lot more kids into this world. So thank you for doing this and building this kind of tool. It's really amazing.
Yeah. Steve, do we have time for one last question from the audience?
Yeah, definitely.
Kevin, do we have the best question?
Yeah, we touched on this a little bit, but John Wester was asking in a follow-up, what role will AI have in aiding visual design? I think you were touching on that a little bit with synthetic biology. and the tools that brings to this enterprise.
Yeah. So one of the things going on right now, if we pull up the surface here, and so I cut out some segments of this, I believe, just to make it nicer on Quest. So this isn't the full ACE2 protein. That's why there's some gaps in the middle. But you could do different color representations. Like right now, this is colored by hydrophobicity. We have a few others for like electrostatic surface potential. But what you could also do, I don't know if you've all played the game Stormland on the Oculus. Stormland, they have a good heads-up display. And so what you do is you kind of like tap your head, and then this AI starts overlaying a bunch of information. So I think what would be really cool is if we could see either on surface coloring or highlighted different segments, basically like what are the areas of interest, or as I'm like selecting things out, and maybe I select from here to there this time, and then I split that out. It could be giving me suggestions on like where to place it. So if we just zoom over to that. Yep. So it could give you ideas on where to place it, what you would like to do with it in terms of like attaching it to an antibody. And so we'd like to see like highlighted information where, okay, if this is going to be my peptide sequence, and I'm just going to import an antibody real quick, or at least the FC domain of an antibody. So it'd be nice if the software was smarter about how it actually told you where to put things. Right now, it's kind of like do things on your own as you see fit, and then run simulations to see if it would work. But we have a few areas that we could potentially attach things to. One over here, these two ends. And so you could have overlaid of information where it says, if I take this guy and let's get rid of that box, and I, attach it over here, is that going to cause something bad? Like, should I add a few different residues in between to help link them together? Or if I deposit this whole thing together, if I link it, can I compare this to existing protein structures and then get overlaid information on what mutations or what modifications I should make to make it better? So AI could have a role on drug design. I actually forgot in the protease scene earlier, we actually had some molecules from in silico medicine that were produced from AI, just generating small molecules. So there's AI generating things for you, but then there's also AI giving you more contextual information and aiding you and helping you become more of the centaur, where it's AI plus human working together.
Are you going to build in the ability for a chemist to teach an AI that this worked, and therefore every time in the future you're trying to design something like that, it'll look for those kinds of patterns and say, hey, there's a potential here?
Yeah, ideally, we'd like to have like a personalized AI. We could start with a base model that's maybe based around like existing chemical compounds and things like that. And then we'd like to see how each person sort of interacts with that. And then there's an interesting model in machine learning called federated machine learning, where each person is sort of training their own model. And then metadata from each model is going to like a global AI that takes all the metadata from all the submodels and then could build better like universal models. And then you could also deploy those universal models back to people. So yeah, they're like, we're going to be going super deep with AI. Right now, we just started with a few things, voice commands, some computational things, and we want full like, I want a fifth person here in addition to the four of us. That is like an AI avatar over here, where we don't ever know if they're an AI or not, but they sound like the smartest scientists in the world, and they're giving us feedback and helping us along the way, or maybe even being an instructor for all the students that are learning this.
And we have a lot of the foundational tools for that already. These simulation software, molecular dynamics allows the AI to kind of close that loop. In a way. So the AI could in the background be running thousands and thousands of different simulations and trying out many, many different combinations. And because the simulation can give you feedback on the stability and aggregation properties, and maybe even in the future immunogenicity of what you're designing, and it can do that far more effectively and with a higher throughput than a human ever could. So I think we already have a lot of the tools to make AI a crucial component of this. We just need to put them together.
Yeah, let's do it. Is that
cool? Yep. So let's go ahead and just place that there. So we have antibody plus peptide chain, and I will take out the camera and put it in a selfie mode. There we go. Oh, John, you were good how you were. And
me.
Oh, it's probably me. Probably me. Okay.
All right, ready. I'm gonna put on the timer. 3.
That's so cool.
3, 2, 1. Perfect.
Awesome.
That was amazing. Well,
awesome.
Amazing time we live in. Thank you to Robert Scoble for joining us to give us a future of what VR is going to look like. Congratulations on your
forthcoming
book.
This is the future. You know, this is pretty mind-blowing example of how new technologies are letting us do things together and see things in a new way. So thank you so much for this. This is really pretty
Absolutely.
Cool.
Thanks to Rob Rhinehart and the Coronavirus Project, Coronavirus Initiative. If you want to get in touch, then you can find Rob through that. And Steve, you've created an amazing immersive visual environment that's going to help rid the world of this horrible virus. So Steve McCloskey, the founder and the CEO of Nanome, thank you very much too.
Thanks.
Cool. Yeah. And thanks Scoble, thanks Rob, thanks Sean, and Philip. Hopefully you see this later on the stream. But yeah, this has been great. I feel like virtual reality is a future that's being accelerated due to the coronavirus, and probably going to be seeing a lot more of this.
Yeah, and we're going to be doing a whole bunch of more live events, and you can find them on our YouTube channel. You can also subscribe to our weekly newsletter at SynBioBeta.com. So thank you all for joining us today, and we'll see you soon. Have a great day.
Cool. Thanks, everyone.
Thank you.
Cheers.